Welcome, everyone. Good morning. We're going to get started. Find a seat, grab some food, coffee. Can we make sure people on Zoom are getting audio? Yeah, good. I was asked to ask people on Zoom when you log in, pin the MiSBIE screen so you have this on screen. Thank you so much for being here. This is really special. I'm so glad this is finally happening. We started MiSBIE 8 years ago, and I think I've been looking forward to this for at least as long. MiSBIE is probably the word that I said the most often in the last 8 years, and it's occupied a lot of my thinking space I think for very good reasons. And this morning I woke up and I sat down, closed my eyes, and I felt so grateful. I felt so grateful that I was gonna meet all of you today, and that we were gonna sit down as a community to learn and to connect with each other, and then with an energetic understanding of what health is and how mitochondria are involved in this process. Something I saw this morning when I woke up is this. We have a geranium that hadn't bloomed in a long time, and then this morning there was 3 beautiful little flowers that bloomed, which felt very special. And this geranium was a gift by Anna in our group. MiSBIE, as I said, is 8 years in the making, and together, through our thoughts, through our actions, what you decide to do, including like spending this day here with this group, is what shapes the future, right? We're all agents of change. We're all collectively creating this world through our actions and where we put our energy. So I'm really grateful that you're here, flying from many different places and spending this time together. And I have the deep sense that the way we do science is beautiful in many ways and limited also in many ways. And too often, I think academic science ends up being about writing papers for other academics, and then we end up doing science for the scientists, and and not seeing the purpose of science is to help humanity and to help us achieve our full potential. So this symposium is dedicated to this, to connecting with people, to creating community, and to empower each other and us as a community to create a better, healthier, more sustainable future. There's this beautiful saying that says, if you want to go quickly, go alone. And if you want to go far, go together. So this is what MiSBIE has been about. And before I introduce a bit, the specifics of MiSBIE, I ask Caroline to say a few words.
Hello, everyone. I'm so excited to be here with you. My hope for today is that we can review our progress together, but also that we will connect and generate new ideas for leveraging this incredible data set that is MiSBIE. I met Martin in 2015 as I joined the Division of Behavioral Medicine as a postdoc. And through countless conversations, I got convinced that I had to get training in mitochondria biology. As all of you know, Martin's passion for mitochondria is really contagious, and it can be very persuasive. So, and when I joined the MiSBIE team, it was really small. We only had 3 lab members. The wet lab was getting set up. And I was really impressed by the integrative vision of this project. And the ambition as well, the goal was to measure any aspect of psychobiology possible, not only in healthy participants, but also including mitochondrial disease patients over 2 days. We had an acute stress protocol, neuropsychological testing, and psychosocial, comprehensive psychosocial assessment along with mitochondrial phenotyping. To some of us, to some scientists, it could seem a little ambitious, but thanks to Martin's contagious energy, and also, most importantly, to the incredible MiSBIE team, we managed to make that project possible. We recruited 110 participants, including 40 individuals with mitochondrial disease, and we have now more than 10 papers published or in preparation. So, yes, so I really hope today we have so much more work to do on this data set, and I really hope today we can generate new ideas and create new projects from this, and also generate new ideas for new research projects ahead.
Thank you, Caroline. So Caroline joined our group as a postdoc back in 2015, 2016, and then now is an assistant professor and has been co-leading the mitochondrial psychobiology group with me. So thank you for doing this together, making this possible. So this reflects the way also that the science can progress. If we want to do advance in a specific, very specific area of science, typically it's called, incremental innovation, and then you get a team that's really good at one thing, and then you do that one thing. If you want to change how we think and discover completely new things about a topic, what needs to happen is connective thinking, and this is what MiSBIE has been of. It's been a hub for connecting people, different ideas, and to discover new things. This would not have been possible without our funders. The Wharton Fund and the Division of Behavioral Medicine. Richard, who's the chief of the Division of Behavioral Medicine, recruited me 10 years ago. Thank you, Richard, for bringing me here and giving me this opportunity. The Irving Institute here at Columbia funded a pilot grant that allowed us to build a MiSBIE group of investigators together back in 2016. And then NIMH, National Institute of Mental Health, funded an R21, a 2-year grant, and then an R01, a 5-year grant that allowed MiSBIE to be. Doug Meineke is a program officer who's trusted us and empowered us to do this work. And then more recently, Bazooki Group, led by Julie Milder, has empowered us to do more with the data set. So when you have an NIH grant, you can recruit participants, do the work, but then you have this beautiful database and this beautiful biobank, and there's so much more typically that can be done, but then if you run out of of funds, that can't happen. So Bazooki Group has allowed us to do more with this. Little housekeeping before we talk about MiSBIE per se. So about today, the talks are recorded. So if you see something today that you think, oh, I wish I could like rewatch this, or I wish I could share this with my colleague, the talks will be recorded and they'll be available on a new YouTube channel. It's called The Science and Experience of Energy. Right? And it's about connecting the scientific understanding of what energy is and mitochondria, mitochondrial psychobiology, and then recognizing that we are human beings. We are moved by experiences, and the emotions we have and the life experiences we have are really end up being movements of energy. And information is available on our website. I think most of you probably registered through this, so the links and the videos will be posted there. We want to create memories today, and we want to hear from you. So you'll receive an email from Vanessa for an online survey after the symposium, and we'd love to hear your questions, your feedback. If you have questions during the day, you'll have opportunities to talk. We've left a lot of room for discussion and the program. And if you have feedback for the next iterations of the MiSBIE Symposium and more events, please leave them there. We also have Piotrek, who's joining us. He's an amazing photographer, producer, cinematographer. So he's going to be creating content and crystallizing memories of what happens today. And he's going to create also a little video that summarizes what this event is about, and that will be on the channel as well. So who is this community? Who's here today in person and on Zoom? And it's quite remarkable that there are people from all over the place. When we brought the team together and invited some of our collaborators 2 years ago, a year and a half, we had 75 people. as of this morning, I think we have about 550 registered participants, about 90 in person, and over 450 online. And you can see people are from all over the place in North America, including West Coast, East Coast, Canada, all over Europe, India, Kenya, and actually and India, Pakistan, Japan, Australia. And yesterday I posted this map without a pin for Africa, and I thought, you know, next time we need to do a better job at getting people from South America and Africa. And Patrick on X, Twitter, said, I'm going to be there. I'm registered. So thank you, Patrick, for joining from Kenya. Birmingham, thank you. Something that's really wonderful also, you know, in terms of who's here, we have people from different slivers of life and slivers of you know, professions. We have community members, so people who are not trained as scientists, but who care deeply about this, and this is really precious. We have scientists from all stages of career, and we have a department chair and distinguished professors to people who don't have a PhD. And I think that this reminds us that to make a significant impact in the world, you don't need a PhD. There's a lot of beautiful things that people have to offer without this this form of training. We have entrepreneurs, people who are building companies and helping to build the futures. We have administrators who make things happen, and they're so important, and they're parts of our lives. Funders, of course, and clinicians, people who actually see real human beings and are faced with real problems. And it's by bringing all of these people together that I think we can create something really special. In terms of food, we'll have food all day, and for lunch and for evening. We'll have a nice little reception with posters this evening, with nice cheese and charcuterie and whatever other else, with an open bar if you're into this. And we want to have dialogue. We want to have discussion. So after the talks, there'll be time for discussion, and then at the end of the day, you'll see in the program, we have a little session where we'll actually bring our minds together and start to come up with new ideas of what we could best do to create a better world. This is the MiSBIE team. These are the people that actually did the work, and this is a lot of people. So it was a challenge to to manage and to keep this team funded for several years. But I'm so grateful to everyone who's contributed to this. Some of these beautiful people are here, and some others have moved on to other things. This is the investigators, co-investigators that were either involved in seeding the project early on or have contributed since then. And you'll hear from some of them. MiSBIE Symposium support team, Vanessa. Where's Vanessa? Vanessa is there. She made this happen. Thank you. And Vanessa is our project coordinator. She kind of coordinates everything in the lab and in my life. And there's Mangesh. Where's Mangesh? Mangesh is there. He's our lab manager. Mangesh and Vanessa, amazing team, and they are the reasons why we are sitting here today. And then there's the rest of the team. This is our lab who've contributed to different parts of making this day happen, and from whom you'll hear about today and their talks, and then this evening and their poster. So today's program, and we'll do this quickly, and then we'll jump into why did we start MiSBIE, and and then after, everyone will tell you about what we learned. So Caroline.
So to start the day, Martin will provide first a short introduction to MiSBIE study design and rationale. From there, we'll have a session on brain mitochondria. Then we'll have a 30-minute break. As Martin said, that's probably the most important part, to be able to connect and ask questions. Then after the break, we will have 2 sessions, one on immune cell bioenergetics and one on health indicators in mitochondrial disease. Then we will have a 1-hour break for lunch. In the afternoon, we will have a session on the energetics of acute stress reactivity, before the last session that will discuss future direction and opportunities to collaborate on MiSBIE projects. Finally, we will close the day with a roundtable for the best research idea, and that will be followed by a poster session to continue conversations over cheese and wine. Again, thank you all for being here.
And for the discussion dialogue this evening, there will be a prize. So the best idea will get this wonderful mitochondria, and there are little mitochondria sprinkled all over to remind you of what powers us. Every time you breathe today, it's to feed your mitochondria. And every time you go to the food table, also feeding your mitochondria. So important stuff. So why did we do MiSBIE? We recruited participants. This was the brochure that we developed many years ago now. And we wanted to understand the link between mind, psychosocial processes, subjective experiences, and the biology. And there's 3 decades at least of solid research on this. And one area that hadn't been explored really is the role of mitochondria, the role of energy in mind-body processes. So we recruited participants. We asked them to come spend 2 full days with us and give a lot of themselves. And we had 2 amazing study coordinators who coordinated the MiSBIE study. And the first meaningful thing that the study coordinator told the participants when they showed up is this: we want to understand you as a person. And I've been a research participant in different studies, and I've never felt like this was really about me, and it was either about they wanted my blood or they wanted a picture of my brain or something like this. MiSBIE was really about understanding each person as a person, as holistically as we can. What we do in science with the beautiful tools of reductionism is we look at pieces, right? We look at this protein, or favorite protein like GDF15, for example, or we look at our favorite enzyme in the mitochondria, or you look at your favorite brain region like the hippocampus, or whatever you're into, your default mode network. So we look at pieces and we become so absorbed with this that we lose track of and lose sight of the whole person, which is really what it's all about. So if you think about life and about human beings as holistically as we can, but with a structured framework, this is, I think, a framework that is useful. I think it's both accurate in a way that it's hierarchical and it's, everything is nested within everything else. But it's also structured in a way that makes it tractable. So we can ask questions about mitochondria and how they affect cells. And then we ask questions about cellular functions and behaviors and how that affect organs, organ level functions. And then we can ask questions about organs and measure different pieces like the brain and the heart and so on, and then ask how does that affect the person's behavior, individuals. And then we can ask questions about how people interact with each other, whether they feel socially supported or isolated, and ultimately this is the basis of what we are, what we experience, and what we're able to do as communities, right? There's energy that ripples from your mitochondria to your cells, to your organs, to you as a person, and then ripples out to other human beings with which you connect. So there's information flow that direction, from very small to very broad, but also there's information flow. We know now from the high levels what you experience actually trickles down, changes how your brain works, changes your heart, and changes your cells and changes your mitochondria. So MiSBIE was about bringing good science to these questions, asking how are mitochondria connected to human experiences. In a nutshell, this is the broad question we want to ask: how does energy in general, and mitochondrial biology in particular, influence human psychobiological processes, human experiences, aging, and the expression of mitochondrial diseases? And there's one preclinical study that motivated this. In science, science often progresses step by step. You want to get across this intimidating river of knowledge, or lack of knowledge, is a big gap. And often you need to set a stone, and then another stone, and then another stone. So this was one of the stone, and this is a study from 2015 that I did as a postdoc that showed that if you change the mitochondria in a living creature, mice, if you perturb mitochondria in 4 different ways, the green, the yellow, the blue, and the red mice have different types of mitochondria, mitochondrial disorders, or mitochondrial defects. And what we found is that if you measure the stress response in many different ways, in MiSBIE we have about 3,000 variables, 3,000 things that we measured, states from each participant. Here there was 74, so this is the preliminary step. Across 74, you can see by the color, this is a heat map. Red is higher, green is lower. How are different aspects of the stress response of the physiology of these animals changed in response to stress, induced stress, if they have different mitochondria? And the picture was very clear. If an animal has different mitochondria, it either perceives stress differently or interprets and responds to stress differently. Right? So that positioned mitochondria as a mediator in this stress disease cascade. And the stress disease cascade is a long-standing problem or question, which is this here: how do psychosocial factors, what we experience subjectively in the mind, right? How does that change biology? How does that affect the rate of aging? How does that change the risk for cardiovascular disease, the risk for neurodegenerative diseases? And we know there is a connection there. And the question we've been asking is, could it be that mitochondria, right, and changes in energetic states in the organism actually is the mechanism through which the stress gets transduced or embedded into the biology? So this was the design for MiSBIE. We thought, let's measure mitochondria with as much precision as we can, and then let's measure stress reactivity, disease biomarkers, which as much depth as we can, and then let's measure people's brain responses, brain structure, but also brain responses to stressors, and understand whether the mitochondria can change the brain and can change how stress responses happen. Last year, we published the MiSBIE mother paper. We call it the mother paper because it is kind of the mother of all other papers. And Catherine Kelly, our previous study coordinator, devoted a lot of most of a year working on this paper. And it's meant to be a community of resources on the rationale, the why of the MiSBIE study, the design, the execution, and to facilitate future collaborations, follow-up studies, and to disseminate results. So if you go on the MiSBIE mother paper, if you go on the website for the MiSBIE page, you'll find the mother paper there and the data dictionary, all of the information you might everything you ever want to know about MiSBIE is there about how the study was done and what's available from the study. And today you'll hear about what we've learned from this. This is the paper here, and you see the number of authors. These are the people that contributed either to the doing of MiSBIE, the planning of MiSBIE, and to the early phases of analysis. And these are the key concepts that we tried to capture in MiSBIE. Understanding how exposures, something that happens outside of you, ends up inside of you, inside of your cells, inside of your epigenome. We wanted to capture things dynamically over time. Health is a dynamic state. It's not a thing, right, that you can measure like a protein. Health is a dynamic state, so we need to capture dynamic processes. And health and disease processes manifest across levels of complexity. At the molecular level, at the organellar level, or favorite organelle, and other ones, cellular level, organ, and then multi-organ and level of the experience. In a nutshell, this is what MiSBIE was about. The most important piece here is the center, right? The participants that we wanted to understand as holistically as possible. And we recruited 70 healthy individuals that exhibit the natural spectrum of mitochondrial health. And when we measured the mitochondria in our healthy controls, you find that there's large differences. We all have pretty different types of mitochondria and mitochondrial energy transformation capacity. And then we were so lucky to be here at Columbia, to be working with Michio Hirano, who's the clinician behind the MiSBIE study. And we were able to recruit and have 40 participants who are clinically diagnosed with the mitochondrial disorder. And some of you, patients with mitochondrial disease, I think are on this call on Zoom, so I appreciate you joining us today and you specifically trusting us for this study. 25 of those have a mutation called the 3243 mutation. In the most severe form, it causes MELAS, mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes. We had 5 participants diagnosed with MELAS and 20 without MELAS, and then 15 individuals with single large-scale mitochondrial DNA deletions, which is basically the piece of mitochondrial DNA that we all have that come from our mother. Right? We all have our mom's mitochondria and mitochondrial genome. Some people have a segment of the mitochondrial DNA missing, which means there's a few genes there that are not present, and then therefore the mitochondria have a hard time fluxing energy and transforming energy. So with those 40 individuals, it allowed us to understand more specifically, more mechanistically, if the mitochondria don't work properly, how does that change the experience, right? How does that change your ability to respond to a stressor, to mobilize cortisol, and so on. And then what you see on the top, what you see at the bottom is what you're going to hear about more today. So I won't go into details, but we measured a lot of things in each participant. And there are three main components. First, we tried to profile things at rest, at baseline, understanding why people are different and how people are different. This is point number one. Point number two was a pretty intensive stress reactivity protocol where we challenge individuals and ask, how do they respond to this? And then the third piece was when they went home for a whole week after they collected saliva samples and physiological measures, behavioral measures. We looked at how they sleep and so on. So those data are still under analysis. So this summarizes or sets the table for today. And what you'll hear about now is the beginning of the scientific program. Anna Manzel, who's a research scientist in our group, is going to chair the first session. So Anna.